Okay, we're going to talk today about predation, food webs, and a variety of species interactions and how we can measure these things in the environment. So there's lots of different ways that species can interact. Herbivory, we're going to go through all of these things in more detail, but in general, we can talk about herbivory, detritivory, omnivory, predation, competition, and a variety of symbioses. So first of all, herbivory. These are animals that feed on primary producers, so green plants, algae, phytoplankton, periphyton, plant leaves, plant stems, plant roots, fruits, mosses, bryophytes, liverworts. The picture at the top is a flannel-mouthed sucker from the Grand Canyon. It's a scraper, so it scrapes algae, and look at those lips. So great. And then this little fish is a gizzard shad. It doesn't look like it would be an herbivore necessarily, but it actually can use mucus and trap phytoplankton on its gill rakers, and then it can eat them that way. Here's an invertebrate grazer or an herbivore. So these grazers can consume algae. They can increase or decrease the productivity of the algal community. So in general, they reduce the biomass, but sometimes that promotes the growth. And so they actually produce more. So productivity goes up. If they graze things almost to extinction, then of course productivity would go down. They can selectively feed on different types of algae, which can change the taxonomic community of the algae on stream bottoms. They can change the form and structure of algal communities. So by taking selectively, say, maybe filamentous forms or other types of forms, they can change how that community is structured. They can alter species richness and diversity of the algae. They can disrupt algal mats, send them downstream. They can alter the nutrient concentrations of streams through their algal grazing, and they can export algal cells downstream too for collector filters, collector gatherers. And we talk about when a new rock is introduced to a stream, there's a succession of colonization by algae, and so grazers can come in and they can alter those successional trajectories. Macrophytes are typically grazed by organisms like crayfish, moose, water buffalo, manatee, hippo, beaver, snails. And there's actually, macrophytes are really heavily consumed. So 5 to 10 times more macrophyte tissue is consumed per area than the plant tissue that's consumed on land. So it partly is because macrophytes tend to not have as much structural, like woody material, and so they're just easier to consume, most likely. Here is a picture of an advancing herd of grazers. These are some Blepharisauridae fly larvae grazing on algae on rocks. You can see they are just cleaning it up. All right, so moving on to detritivory, so detritus. This is kind of an important point. So we talk about autochthonous versus allochthonous, or allochthonous versus autochthonous in this case. So autochthonous energy is energy that's produced within a system. So if you're thinking about a stream, then algae would be autochthonous, right? It's growing from within the stream. But the stream is also getting a lot of important energy from outside of the stream. Those are called allochthonous inputs. And so things like leaf litter and woody debris and twigs falling into the stream from the forest, those would all be allochthonous inputs. And those are some of the things that detritivores are going after. They are most likely going after the microbes that colonize these materials. So leaf litter is an important food resource in streams. The shredding invertebrates are typically going after the microbes that have colonized the leaf litter and the wood. So things like fungi and bacteria that are growing on the leaf surface. Detritivores can influence dissolved organic carbon concentrations, and so can these allochthonous inputs. So when leaf litter falls into the stream, dissolved organic carbon can leach out. Soil organic matter can come into stream systems through leaching and runoff from the forest floor. And detritivores, like I said, might be trying to eat the microbes and inadvertently eat the wood. Some organisms will ingest a lot of material that they can't digest. So, you know, when you're looking at stomach contents, like at the bottom there, you might see a lot of bits and pieces that the organism would just expel without actually digesting, and some things that they might actually digest. So this is one of the reasons that stomach content analysis is really interesting, but it can kind of sometimes give spurious results. And then a lot of omnivores will end up consuming some detritus as part of what they eat. So detritivores, what's interesting is that detritus is not very nutritious. It tends to have lots of big recalcitrant compounds like lignins, but the microbes that colonize these materials can increase its nutrient content and help break it down and make it more available. So detritivores convert a large proportion of available energy into production, a larger proportion than plant-based systems. So detritivores are really super important, both terrestrially and in aquatic systems. Energy is more conserved and more recycled in what we call brown food webs that are driven by detritus, than green food webs, those food webs that are driven by plants. And there are higher consumption efficiencies. So basically what that means is organisms, when they eat something, they eat all of it, right? There's a lot less waste in detritivore systems. They also assimilate more of that material into their body. So they have higher assimilation efficiencies, partly because of the microbial community that's helping there. And production efficiencies. So they produce more per unit of food that's eaten. So overall, there are higher trophic efficiencies in detrital systems. This is one of the reasons that I like to study detrital systems. There's also a high degree of detritivory. So even when you're looking at an organism that you might consider, you know, a collector-gatherer or a grazer, they're probably also eating a lot of detritus. And so it just kind of makes food webs really messy in a sense. Detritivores are mainly eating the microbes. So I think of it like a dry, boring cracker, right? But then it tastes much tastier if you put a lot of peanut butter on it. So the microbes are like the peanut butter growing on the cracker, and then the detritivore eats the whole thing. They're eating the peanut butter, eating the cracker, and getting a nutritious meal. So litter quality, this organic material, is determined by a whole variety of factors. I will talk about it in other lectures. But things like elevated carbon dioxide in the atmosphere and other environmental conditions can change litter quality, sometimes making it harder to break down. Okay, so many organisms are omnivores, but maybe just for part of their life cycle. And omnivory, kind of like detritivory, what I showed you there, complicates food web analysis. So strong omnivory can destabilize food webs, whereas weak omnivory can stabilize them. And omnivores tend to be really good invasive species because they can eat all kinds of different things. Food webs are not this simple. So we used to talk about food chains more, and this is a little plush animal toy called the Food Chain Friends. Things are not this simple. Food webs are much more complex than this and require some really interesting ecological tools to study. Okay, moving on to predation. Predation is ubiquitous. There's all different kinds of predation, vertebrate on vertebrate, like you see at the top, invertebrate on invertebrate, like you see on the bottom, but vertebrates can eat invertebrates and invertebrates can eat vertebrates, all kinds of predation out there. There are lots of really cool anti-predator traits. So how do organisms protect themselves from predators? So prey can develop mechanical, chemical, and or behavioral protections. So things like protective armor and spines, where there are a lot of predaceous fish, then the zooplankton in the system will grow these long spines like you can see in the pictures. They can produce toxins, so prey might produce toxins, or they might produce just low quality biomass. They can also move around to avoid predators. And so the kind of colorful figure at the bottom shows you a time series from 5 a.m. through noon on to 1 in the morning, basically a full 24-hour period. And the colors, so the the dark purple color is the highest biomass of these little Daphnia, these little water fleas. And so what you can see is that right about 5 o'clock in the morning when the sun rises, all of the zooplankton drop down below into the aphotic zone. So they get out of the light so that they can avoid predators. And then right around 6 o'clock in the crepuscular kind of twilight hours, they start rising back up and they spend their time up high grazing on phytoplankton in the photic zone at night. So this is called diel migration. And it's a kind of a vertical migration in the water column. Another thing that prey can do would be to school, or things like synchronous hatching for mayflies can provide just like a whole bunch of organisms all in one place, which means that any one organism is protected from predation because of all of its friends around it. Organisms can also sense chemical indicators of predators in their environment, and they can avoid them, try to avoid them. Some of these traits are inducible, which is really cool. And that means that when they sense a chemical indicator of a predator, then they might grow spines or they might produce offspring with longer spines. And so those would be inducible traits. A lot of plants can induce defense chemistry in their leaves. Say they flush another set of leaves that has higher defensive compounds against herbivores. So those would be inducible versus constitutive traits. What kinds of things can predators do? Well, they need to encounter and detect their prey. They need to attack and capture it, and then they need to ingest it somehow. So there's a theory called the optimal foraging theory that basically says that when food is scarce, anything will do, but when food is abundant, predators are more selective. So I like to think about my daughter's Halloween candy, and when it's abundant, right at the day after Halloween, I can selectively forage for my favorite Halloween candy. But at the end of a few weeks after everything's kind of been picked apart, I will take whatever is left, right? Then you're just like, where is it chocolate? Yes, I'll eat it. All right, so there are sit and wait predators. They have explosive lunges. They have hinged jaws like this odonate in the figure up at the top. They might use stinging nematocysts or electrical currents like eels. Carnivorous plants have a variety of tactics. Some of them are more active and grasping, and some are more passive and sitting and waiting, right? Predators also use visual, tactile, mechanical, and chemical cues to detect prey. Some predators are gape limited, meaning they need to consume their prey whole, and so they're limited in terms of how much they can get their mouth around the prey. So if their prey item is too large, then they won't be able to attack it.